Welcome! In this video tutorial, I will briefly describe the output possibilities of the Advanced Cell Classifier's regression plane. To obtain output from the regression plane, the user must have previously defined classes of interest with at least one regression class, some annotated cells in them, and a trained classifier. For the sake of clarity, classifier denotes the model trained to classify the cells in different classes, including the regression ones, whilst regressor is the model trained to define the position in the regression plane. The main aim of the regression plane is to predict cells in a continuous manner, without the need of fixing intermediate cell stages. This way, a biological process is considered inherently continuous. Each cell predicted by the classifier as belonging to a regression class will be also associated with a position in the regression plane by the regressor. By enabling cell classes and clicking on the Predict Image button, the regression values of each selected cell can be viewed. To automatically predict the position of cells from a set of images, simply use the Predict Images button in the regression plane. The cells of the selected images will be automatically located on the regression plane. All the cells that belong to the training set will be displayed with blue border. Additionally, it is possible to visualize a selected feature's value distribution by pressing the Color Frame button. After selecting the feature, adjusting the color settings, and pressing the Apply button, the border of each cell becomes highlighted with the color associated with the feature value. In order to run a regression analysis of an entire plate or a longer list of selected plates, click on the Predict Plate button. In the opening GUI, the heat map resolution setting specifies the number of bins when representing the regression plane as a discretized heat map. The output files will be automatically saved in a folder named regresult in the path where your data is placed. Then, from an intuitive plate and well selector, it is possible to select the plates to be analyzed and the wells to be considered as control for the analysis. During the analysis, you will be asked if you would like to perform optional, unsupervised analysis of your data to help the exploration of the outcomes. To maximally exploit these techniques, you need a plate layout file. A plate layout file is a simple, comma-separated value file with two columns, the ID of the well and the name of the treatment in each row. If you don't have a plate layout, it will be generated automatically. Several files will be automatically generated in the result folder. The main output of the regression plane is the regression report PDF file, summarizing the predicted positions of each well in the form of a heat map. The heat map represents how the cells coming from that particular well were placed on the regression plane. A brighter region means that more cells were placed in that region of the regression plane. The output files of the unsupervised analysis will be placed next to the regression report PDF. Plot of plots is a visualization method to help users discover treatment similarities. Heat maps displayed in a similar position highlight treatments that have similar effects on the cells. If you have a plate layout, then the replicates are highlighted with the same color. Besides visualization in the plot of plots, hierarchical clustering of the treatments can also be performed. A clustergram is reported in the treatment comparison _ distances.pdf file. This provides an alternative way to compare different treatments. The grid in this figure was created by using colors to visualize the distances between the treatments. The dendrogram represents the connections between different treatments. Similar treatments are connected with shorter lines. Finally, In case your dataset is a sequence of images of the same cells acquired at different time points, for example live cell imaging, there is also an opportunity to visualize their dynamics, meant as the change of their positions over time. Trajectory plot is the module to generate a trajectory for each cell, showing the xy position of the same cell in the regression plane at different time points. You can start the module by clicking on the Plot Trajectories icon in the toolbar. You will be asked for a single-cell regression prediction file and a tracking file. The former one, the single-cell prediction file, is automatically generated in the output directory when predicting a full plate. The latter one, tracking file, is a simple comma-separated value file that defines the trajectories by unique track IDs. The cell instances composing a given trajectory are defined by their plate and image names, frame numbers, and their positions in the image. In case you do not have access to a tracking file, it is possible to generate it through a built-in wizard. After clicking on the Start button, the trajectories are visualized. The trajectory plot provides several options to analyze the dynamics of the data by grouping, filtering, and animating the trajectories. You have now seen the main output possibilities of the regression plane. To learn about other output possibilities regression plane can offer, please read the help documentation file provided with the software. Thank you for your attention.